The cell cycle is a process by which a cell divides. Such a process is necessary for reproduction, growth and development, and tissue renewal. The cell cycle can be broken down into two processes, interphase and mitosis. Interphase, the longest phase of the entire mitotic cell cycle, includes three subphases, G1, S, and G2. G1 is simply a phase preparing the cell for S phase. S phase is the process in which the chromosomes and the centrosomes of the cell replicate. G2 is the phase preparing for M phase, or mitosis. As denoted by the cheese, the chromatin of the cell is in relaxed form in all three of these phases. Mitosis can be broken down into four subphases: prophase, metaphase, anaphase, and telophase, and cytokinesis. In prophase, the chromatin fibers take on a tighter, more solid form. The nucleus, or garlic bread, has now disappeared, and the duplicated chromosomes now appear as two identical sister chromatids joined at their centromeres. The mitotic spindle, depicted by the spaghetti in this case, are connected to two meatballs, which represent the centrosomes. In the beginning of metaphase, the nuclear envelope disintegrates, and the microtubules enter the nuclear area and attach to the more condensed chromosomes. As metaphase continues, the centrosomal meatballs now make their way to opposite sides of the cell, and the chromosomes align along the metaphase plate. Now all sister chromatids are connected to kinetochores on opposite poles. Metaphase is the longest phase of mitosis. Conversely, anaphase is the shortest phase, and it begins when the proteins holding the sister chromatids are separated. This means that each chromatid is now a chromosome. The chromosomes begin moving to opposite ends of the cell, and while this is going on, the cell begins to elongate. By the end of anaphase, both sides have the same amount of chromosomes. In telophase, daughter nuclei form in each cell, and the nuclear envelopes begin to reappear. The chromosomes begin to become less condensed, and in cytokinesis, the division of the cytoplasm is finished. In animal cells, a cleavage furrow, using actin and myosin, forms, pinching the cell into two independent cells. We began the mitotic process with a cell containing 4 chromosomes and finished with 2 cells containing 4 chromosomes. We now have spaghetti dinner for 2.
Meiosis is the process by which gametes or sex chromosomes are developed. It is important to growth, repair, and replacement. It involves 2 sets of processes, meiosis I, which separates homologous chromosomes, and meiosis II, which separates sister chromatids. Meiosis I begins with prophase I. In prophase I, instead of having the replicated sister chromatids, we have replicated homologous chromosomes. This is denoted by the orange and white cheese. Crossing over occurs now, and it is while homologs are in synapsis. Each homologous pair has one or more chiasmata, the site at which crossing over between sister chromatid occurs. Like in mitotic prophase, centrosomal movement, spindle formation, and the breakdown of nuclear envelope occur. In metaphase I, the homologous chromosomes are arranged on the metaphase plate, with 1 chromosome on each pair facing each side. Each pair of homologous chromosomes is attached to kinetochore. In anaphase I, the homologs separate and move toward opposite poles, but the sister chromatids remain together. In telophase I, with equal amounts of replicated chromosomes on either side, the cell begins to separate. Meanwhile, during cytokinesis, 2 haploid daughter cells are created. Meiosis II. In prophase II, chromosomes of 2 chromatids associate at the centromere, much like in mitotic prophase, but here it is happening in 2 cells. In metaphase II, chromosomes align along the metaphase plate and are attached to the kinetochore on opposite poles. In anaphase II, the proteins holding together sister chromatids break down at the centromere and the chromosomes separate. The individual chromosomes begin moving to opposite poles. In telophase II, the nuclei form in each cell and the chromosomes begin to relax. As cytokinesis II occurs, 4 haploid daughter cells with genetically distinct chromosomes are now formed, as opposed to the 2 diploid cells formed in mitosis. We reduce our number of chromosomes by half. We now have spaghetti dinner for 4.